Namaste viewers, this is Dr. Pratibha Case. Today I'm before you to present first semester BSc Biotechnology Cell Division Mitosis. Okay, before I begin, I request you to subscribe this channel to support. Thank you. I hope students are making note of all the online classes in your notebook with the date and time. Now let us go through the cell division. Okay, where do cells come from? You can question yourself before understanding about the mitosis. You can question yourself, where do actually the cells come from? Sometimes you accidentally bite your lip or skin or your knee, and any damage may occur to the skin, right? But in a matter of span of days, the wound, that particular wound heals, right? Is it a magic, or is there any other explanation? We need to think about this, right? Every day, every hour, every second, one of the most important events in life is going on in your body, right? It means, what is that? That is, cells are dividing, right? The cells are divide. Cells are dividing. When cells divide, they make new cells, right? A single cell divides to make two cells, and these two cells then divide to make four cells, and so on and so forth, right? And we existed also through one single cell, right? Zygote, which is the fertilization of male gamete and the female gamete. That one zygote, that one particular single cell, that single cell, now it has divided, and now we are human being, right? A gigantic human being, we can say, right? Single cell, so many things such as happen. We call this process cell division and cell reproduction, right? Because new cells are formed when old cells divide. The ability of cells to divide is unique for living organisms. Why do cells divide? Can question yourself. Cells divide for many reasons. For example, when you bite your skin or your knee or lips, or during any damage, cells divide to replace old, dead, or damaged cells, right? Cells also divide so living things can grow. When organisms grow, it isn't because cells are getting larger, okay? Organisms grow because cells are dividing to produce more and more cells. In human bodies, nearly 2 trillion cells divide every day, okay? Now let us go through here. How do cells know when to divide? In cell division, the cell that is dividing is called the parent cell. The parent cell divides into two daughter cells. The process then repeats in what is called the cell cycle. In my another video, we'll go in detail about the cell cycle, different phases. Okay. Can make out here the so how does actually exactly cell divides from the single cell to the multiple cells. Okay. Next, how cells divide. Depending on the type of cell, there are two ways of cell division. What are those? Mitosis and meiosis. Of these methods, cell division has special characteristics, can be seen in the mitosis. It is a process of cell division in which chromosomes replicate and get equally distributed into two daughter cells, so that these cells have the same number and type of chromosomes as are present in the parent cell. So it is also called as equational division. Okay. It is also called as somatic division because it is a division of somatic cells. Plant and animal cells both undergo mitotic cell divisions. Their main difference is how they form the daughter cells during cytokinesis. During that stage, animal cells form furrow or cleavage that gives way to the formation of daughter cells. Due to the existence of the rigid cell wall, plant cells do not form a furrows. Okay, because that's the main difference between the plant cell and the animal cell. In the plant cell, you could find the presence of the cell wall, whereas in the animal cell, you don't find the presence of cell wall. Okay, mitosis was for the first time, which was demonstrated by Strasburger, okay, in the year 1870 in plant cells. The term mitosis was the first time which was coined by Fleming. Mitosis has four important stages: prophase, metaphase, anaphase, telophase. Hope you're making note of all this information. Division of body or somatic cells is known as mitosis, right? It is a complex process involving a series of changes leading to the formation of two equal daughter cells from a single parent cell. It was observed and named by Fleming for the first time. It is also called somatic cell division and occurs in all body cells. It is called equational division because the daughter cells have equal number of chromosomes, whereas the word somatics, which stands for, that is body chromosomes or body chromosomes. Okay. Now let's go through here karyokinesis. The replication and distribution of chromosomes are known as karyokinesis. That is related with the chromosomes. Cytokinesis, division of cytoplasm into these two separate cells, that is related with the cytoplasm. Cytokinesis, the chromosomes replication and distribution, karyokinesis. Okay, now let us go through in the mitosis stages. First one, that is interphase. After that, prophase will come. Okay, this is the period between two mitotic divisions where both nucleus and cytoplasm attain maximum growth. The following process occur during the interphase: synthesis of DNA and protein, DNA replication leading to chromosome duplication, building up of ATP, which is used for different stages of mitosis. Then some important features that can be identified during this stage are nucleus and nucleolus appear conspicuously large, nuclear membrane is intact, the duplicate chromosomes are visible as extremely thin strands. Okay, this is all about the interphase. Okay, you can see here the structural representation of the interphase of the animal cell. Okay, you must also know here the difference between the animal cell and the plant cell structure. Whenever we write this round, no, this will be the animal cell. Whenever plant cells we write, no, that will be always rectangle. And in the plant cells, you won't find this particular centromeres, asters, you won't be able to find. As you can see in the interphase, the appearance can be seen nucleolus, nuclear envelope, then plasma membrane is here, you can see. And in the animal cell specifically, you can see centrosomes with centrioles, asters can be seen. In the center, you can see the chromatin bodies. Okay. Next, now let us go through the stage 1, that is prophase. Okay. In this prophase stage, the chromatin material condenses into distinct chromatin threads by dehydration. Okay. The chromatin threads gradually become shorter and thick due to the specialization and dehydration. Each chromosome is formed of two sister chromatids, which are held together along their length by centromere. The nucleolus and nuclear membrane disappear by the end of prophase. The centrioles duplicate and move apart to occupy the opposite poles of the cell. Numerous microtubules appear around the centrioles to form a star-like aster. The microtubules appear across the cell from the aster to form spindle fibers. Okay, so you can see this is the animal prophase stage and this is the plant cell prophase stage. You can see the difference here. This is circular, right? And this is the rectangle in the plant cell. You can see here the prophase diagram. Okay, centrioles, formation of the mitotic spindle fibers. Then you can see the chromosome consisting of two sister chromatids. Then you can see this as the early prophase, and here you can see this is the late prophase. In late prophase, you can see the formed asters and centrioles, everything can be seen here. Okay, and you can also see fragments of nuclear envelope here, right? And here you can see the spindle pole. Then here you can see the centromere. Okay, that is about the prophase diagram. Now let us go through metaphase. In metaphase, what happens? All the chromosomes which are aligned in the center. Okay, metaphase is identified by fully formed fibers. Okay, chromosomes. Due to heavy coiling and dehydration, the chromosomes become short, thick, okay, and thick. Then the two chromatids separate except at the centromere. All chromosomes move towards the center of the cell and orient themselves on the equator of the spindle. In such a case, the centromere lies in the equator, okay, and the asters are oriented towards the pole. In the animal cell, okay, asters which are centrioles which are oriented towards the poles. The centromere of one chromatin faces one pole and the other face the other pole. Okay, like a you can see here like a V shape exactly. You can see one centromere will be facing one pole, other faces the other pole, and they move slightly apart. You can see this is in the plant cell, the metaphase which are aligned exactly the center, and centromeres also exactly in the center. And here this you can see in the animal cell. Okay, you can see this diagrammatic representation, pencil sketch. This is the metaphase. Now let us go to anaphase. Centromere of each chromosomes divide and allow the separation of the chromatids into two dot chromosomes. What centromeres were so facing the two poles, and the V-shaped pole which is going to be apart. Okay, then the daughter chromosomes move apart and migrate towards opposite poles. Okay, equally. The movement of chromosomes is governed by the constant contraction of the spindle fibers. Equal numbers of chromosomes are present which are placed at opposite poles. Okay, here you can see the presence of chromosome equally they distributed to the each poles, right? see this distribution which is not happening properly means then what happens, we can see different syndromes. Okay, we are studying in karyotype, that is somatic syndromes, right? We have two types of syndrome, that is autosome syndromes and allosome syndromes. Whereas in this particular case, what happens when the chromosomes equally, they won't divide, and one of the whole chromosomes moves to one particular pole. Okay, if one particular pole, what happens, they we can see the changes in the number of the chromosomes. Okay, the what are the collect set of chromosomes must be there, that won't be there. So this anaphase phase is very unique, and which is very important for the daughter to become a daughter chromosomes. Okay, in this case, what has happened here, equal number of chromosomes are present which are placed at opposite poles. Equal number of chromosomes you need to find in both the poles. Moving daughter chromosomes exhibit different shapes depending on the position of the chromosome. Here you can see very beautiful diagrammatic and microscopic view of the anaphase. This is the anaphase. You must get like this when you perform the mitotic experiment by using onion root tips. Okay. Next, which is the next step? Next step is telophase. Okay. Here what happens? The chromosomes reach the opposite poles, right, and form two groups, right? The chromosomes gradually undergo rehydration and despiralization to form chromatin. Now, nuclear membrane is formed, whereas in metaphase and anaphase, nuclear membrane was not there, right? Now we can see the formation of nuclear membrane around each group of chromosomes. What are the chromosomes were there? You can see, take this also as a late anaphase. Okay, during this phase, what happens here? The plasma membrane, the nuclear membrane will start forming. Okay, then each group of chromosomes, the nucleus is reformed, right? And asters and spindle fibers, here asters and spindle fibers break down and are absorbed into cytoplasm. Okay, centrioles at each pole are surrounded by centrospheres to complete the centromere. Okay, you can see this in the particular animal cell, whereas in this case you can see this is in the cell plate formation in the plant cells can be seen. Okay, so you can see the entire structure presentation of the mitotic stages in the plant cell. Interphase, and this is the early prophase, and this is the prophase, and this is the late prophase. You can see the metaphase, then here anaphase, telophase, and the formation of the cell plate, and you can see the cytokinesis. In the beginning, I told you that in cell division, you can find the formation of karyokinesis, right? Karyokinesis, and also you can see the cytokinesis. Okay, in your record, you can write this particular structure representation. Okay, pencil sketch: interphase, prophase, metaphase, anaphase, and telophase. Okay, as this is the plant cell, you can see the cell plate formation, whereas the animal cell, you'll be able to see the cleavage furrow formation. Hope this is clear to you, right? This is the microscopic view of the interphase, then prophase, metaphase, anaphase. This is early telophase, and this is the telophase. Hope I made myself clear to you. Okay, if you have any doubts, you can reach to me at 9844668811. Okay, before concluding, I just want to share with you, you have to be a warrior and you should not be a worrier. Okay, you must fight against bad time, and God helps to those who help themselves. I, Dr. Pratibha K, signing off for today. Stay tuned with me, stay connected. Okay, and I'll be back with some more videos very shortly with meiosis and also the experiment to perform this mitosis and meiosis. Thank you for being with me.